Good afternoon. I'd like to welcome all of you to the second presentation in our 2017 Laureate Lecture Series. For more than a decade, this series has featured talks by some of the most accomplished scientists from across the country and around the world. Today is a good reminder that such scientific excellence exists right here in our own community as we welcome Dr. Subra Suresh, president of Carnegie Mellon University. If you read his guest column in the Post-Gazette yesterday, you already know this, but it bears repeating. Subra came to this country as an immigrant. He arrived as a 21-year-old man from India with a one-way airline ticket purchased on a loan, a half-empty suitcase, and less than $100 in cash. Based on this very inauspicious beginning, nobody could have predicted the outcome. A doctorate from MIT and a stellar career in material science and in engineering. Discoveries that have advanced human knowledge and are helping this nation live up to its promise. And unanimous confirmation by the United States Senate to the role of director of the National Science Foundation. We're fortunate to have Subra here. There are more robust partnerships between Pitt and CMU than I can hope to name, but they include the Center for the Neural Basis of Cognition, which integrates Pitt's and CMU's strengths in basic cognitive and computational neuroscience, the Medical Scientist Training Program, a highly successful dual degree program funded by the National Institutes of Health, through which students earn a medical degree from Pitt and a PhD from a basic science or an engineering department, either at Pitt or at CMU. And a great many other close collaborations between individual labs and investigators, including Subra and members of our own medical school faculty. It's fitting then that Dr. Suresh recently added a University of Pittsburgh title to his CV. As of 2016, he is our medical school's 21st century professor of biomechanics in medicine. Subra's research epitomizes interdisciplinary science in that it spans the fields of engineering, technology, and biomedicine. He's an innovator in materials design who also explores the nanomechanics of biological cells and human disease. Subra earned his bachelor's in mechanical engineering from the Indian Institute of Technology in Madras. He received his master's in mechanical engineering from Iowa State University, and he earned a doctorate at MIT in just 2 years. He completed postdoctoral research at the University of California, Berkeley, and the Lawrence Berkeley National Laboratory. He began his independent academic career at Brown University, where he was honored by the White House and the National Science Foundation with the Presidential Young Investigator Award at the age of 29. He joined the faculty of MIT's Material Science and Engineering Department in 1993. He became head of that department in 2000, and he was named Dean of MIT's School of Engineering in 2007. In 2010, Dr. Suresh became director of the National Science Foundation. Upon his departure from the NSF 3 years later, President Obama described him as a consummate scientist and engineer, beholden to evidence and committed to upholding the very highest scientific standards. As CMU president, Subra now directs one of the world's leading educational and research institutions, whose scientific achievements are internationally renowned. But he has earned many honors himself, and he is one of only 19 Americans with membership in all 3 of the national academies: the National Academy of Sciences, the National Academy of Medicine, and the National Academy of Engineering. But today, it is our honor to host him for this lecture. Please join me now in welcoming Dr. Subra Suresh to deliver his talk, Study of Human Diseases at the Intersections of Engineering, Sciences, and Medicine.
Thank you, Dean Levine, for your very kind introduction. It's a great honor and a privilege to come and have this opportunity to present a technical lecture at the medical school at the University of Pittsburgh. Even before I came to Pittsburgh 3 years ago, I knew quite well how celebrated this institution is. And after coming to Pittsburgh, I've had the extraordinary pleasure of working closely with a number of colleagues from the University of Pittsburgh, as well as from the Pitt Medical School. I would particularly like to single out two people with whom I've developed very close collaborations in the last 3-4 years. One is Dr. Greg Cato, hematologist at UPMC and the Pitt Medical School, who's here, and also Dr. Yoel Sadowski, who's here, who's the head of the McGee Women's Research Institute in Pittsburgh. It's been a great privilege to be part of this. And I'm also very grateful to Art Levine and to Chancellor Gallagher for their not only strong desire for collaboration between the two institutions, but also their warmth and welcoming me, not just to Pittsburgh, but also to the broader intellectual community of Pittsburgh, of which University of Pittsburgh plays a major role. So thank you very much. I've been extraordinarily fortunate to benefit from collaboration with a large number of people you will see, and especially in light of the angst at pretty much all American universities these days with respect to the talent pool that may be impacted either in reality or in perception by some of the directions in which we are heading. One of the things you'll see names of people who have contributed to what I'm going to talk about. I'm just presenting all of their collective work, and without them, this work would not have been possible. I would also like to start by singling out one individual who has been part of every piece of work that I'm going to present for the last 15 years that has been done in my research group, I'm very glad he's here with us today, and that's Dr. Ming Dao, who's sitting here, who's been a collaborator in this work for many years. And when I was at the NSF, he ran my research group for 3 years. So thank you, Ming, for coming. What I would like to do is to start with a question. As Art mentioned in his introduction, I'm an engineer by training, so many of you, especially a number of people in clinical medicine are probably wondering, what are you doing here? And so as a wanderer into the field of medicine, about 15 years ago, we asked the following question. My background is mechanical engineering, and over time, my work moved from macroscopic properties of materials to nanoscale properties of materials. So in 2002, when I was head of the Department of Material Science and Engineering, we created a nano lab, and I had a colleague, a biologist, a very celebrated biologist who came to that event, and he said, why are you working in dead materials? You should be working in living materials. And I thought that was a very insightful suggestion. And then a few months later when I met him, he said, you're not going to excite me with all the fancy stuff unless you show me what impact it has on human health or disease. So I thought that was a very insightful comment. So taking his criticism or comment into account, that's what steered us in this direction. So we asked the question, how can I bring my expertise from a very different field, especially to human diseases? Fortunately, we were also at the right place at the right time. For example, you know that in 2000, President Clinton launched the National Nanotechnology Initiative at Caltech. And we had one of the very first nano centers that I was lead PI of. So how to apply nanotechnology? The Human Genome Project was in full swing in the early 2000s. So gene modification, knocking out of specific genes, what impact does it play? Experimental nano tools enabled us to take single biological cells and molecules and probe them in ways in which we could not have done just a few years earlier. In parallel with all of this, we had major revolutions in computation and computational biology, both with respect to hardware and especially with respect to software, and more recently into mobile devices. So if you put all of this together, all of this was accelerating significantly in the early part of the 21st century, about 10, 15 years ago. That's when we started this work. So we asked a fundamental question, the motivation for this work. Two related questions, both forward and reverse problems. How do properties of cells, this is what engineers focus on, this is what material scientists focus on. The material science is a field that is the interplay among making materials or processing, properties, performance, and function, and microstructure. And taking the same paradigm to biological cells and molecules, what contributions can we make, especially at the cell and molecular level? So that's the question. So the first question in the forward problem is, how do changes in cell properties influence diseases? The reverse problem is, how do diseases influence cell properties? So we have origins of diseases that we don't know about, such as cancer. We know origins of certain diseases, like infectious diseases, such as Plasmodium falciparum malaria. We have diseases where we know the precise genetic origin, such as sickle cell anemia. And in the sixth position of the amino acid, if there is a defect, you get a sickle cell trait or a sickle cell disease. So given that broad spectrum of things, can we pick broad classes of diseases where we can focus on at least a partial answer to these questions and use computational tools, the latest tools in nanotechnology and the latest tools in experimental work across disciplines? Can we bring them together? And in each of these topics, we absolutely make sure that we have a bona fide biologist and a bona fide medical doctor or a scientist who can keep us honest and keep us straight. So the goal would be, of course, to understand nature. That's the first major goal. But of course, can we use that to improve diagnostics, therapeutics, and drug efficacy assays? So we have done a lot of different things. I'm going to focus on three broad areas, not necessarily in this order. Different types of human cancer. At the cell level, how can we bring the latest tools from physics, from biology, from material science, from different branches of engineering, electrical engineering, microfabrication, et cetera, computational biology, to improve our understanding of disease diagnostics. Infectious disease, so cancer as one major class. We've looked at different types of cancer. Plasmodium falciparum malaria, the most lethal form of malaria. So the two common variants of the plasmodium for humans is Plasmodium falciparum and Plasmodium vivax malaria, where falciparum causes most of the mortality. Currently, several hundred people per year die from malaria, mostly children with underdeveloped immune system. And the two most common forms of malaria are placental malaria and cerebral malaria. Hereditary blood disorders, and especially genetic factors influencing cell-level properties and how they influence disease state, pain management, and therapeutics. Sickle cell disease is a very good example of this. So these are three case studies that I would like to take to walk you through, so that the talk is not just very general at a high level, but there is also, you can see some of the potential implications of the work. So I'm going to start with first a video. There'll be a series of videos. The first video, not yet. Thank you, Ming. The first video, I want to give a little bit of introduction. Let's take cerebral malaria as an example. Why is mechanical engineering important? So every second, our bone marrow produces hundreds of thousands of red blood cells. A red blood cell, a human red blood cell, is about 8 micrometers in diameter. It's a disc-shaped. 8 micrometers is 1/10 of the thickness of the human hair, to scale it. So if you take a piece of human hair, 1/10 of the thickness is the large diameter of one red blood cell. The smaller diameter is 2 micrometers. And the thickness of the membrane is a few nanometers. And inside that, you have hemoglobin. And it doesn't have a nucleus, and it has a two-dimensional cytoskeleton. And so it's the simplest form from an engineer's perspective to model. And 42% of the volume of blood in our bodies is red blood cells. That's why the color of blood is red. So where does engineering come in? This 1/10 of a human hair thickness red blood cell, as it navigates through the body to deliver oxygen to distant parts of the body and take carbon dioxide back to the lungs, it has to navigate through small openings in the body. Two of the smallest openings are the small blood vessels in the brain, whose inner diameter is as small as 2 to 3 micrometers. And the smallest fitness test for the red blood cell in the human body is in our spleen. The interendothelial slit has a very small diameter, very small opening, and the red blood cell has to squeeze through this. That's the fitness test for the cell. So an 8 micrometer disc has to squeeze through a 2 to 3 micrometer tube to deliver oxygen and take back carbon dioxide. And it has to do it millions of times during its course of survival in the human body over a period of 120 days. This is what mechanical engineers call fatigue, back and forth stretching and going back. The cell stretches by more than 100% and come back. And this is not new in the medical community. In fact, if you go to the malaria literature, since the 1980s, microbiologists and parasitologists have talked about deformability. So the reason we decided to get started in this was, if parasitologists can talk about deformability, somebody who does deformability for a living should be able to talk about parasitology. So go in the reverse direction. And that's what brought us to this problem in the first place. So what I'm going to do, despite all the advances in medicine, we still don't have visualization tools. As a red blood cell, which is a disc shape, goes through a small blood vessel in the brain, how does it deform? So if you look at textbooks in biomedical engineering, they will say a disc-shaped red blood cell takes the shape of a bullet as it goes through a flexible tube in the human body, and then when it comes out, it springs back to its initial shape. So the first video that I'm going to show is an in vitro demonstration of how a live human red blood cell will navigate through a small blood vessel in the brain. It's an in vitro demonstration. It doesn't have the right chemistry, it doesn't have the right biology, but it has the right geometry, and it has the right mechanics without the right chemistry. So this is the work of a former graduate PhD student of mine, David Quinn, who's here, who's now at CMU, and this is part of his PhD thesis work. Thank you. So before you click it, one more thing. So as you see on your left-hand side, so this is blood, human blood, which we used to obtain every week from the blood bank, tested for all the disease and all the protocols. You send it through a microfluidic device made of a polymer called PDMS, which has an inner cross-section, which is exactly the same as the cross-section of a blood vessel in the human brain. And what you're going to see is a disc-shaped red blood cell, as it squeezes through the tube, a narrow tube, takes the shape of a bullet. When it comes out at the other end, it springs back to its original shape. This is what our red blood cells do repeatedly, millions of times during 120 days in the body. That's what you see in the video. This is a high-speed camera. We can slow this down. So if you're a mechanical engineer, there is a million more pieces of information in this that doesn't immediately meet the eye. For example, you can slow down the exit. You can do a viscoelastic modeling. You can actually take the time it takes. You can look at the time it takes for the red blood cell to spring back to its shape. You can calculate the characteristic time of relaxation, which for a human red blood cell is 300 milliseconds. So if you know the time frame, you can verify that through independent techniques, so you can calibrate your experiments. So there are many different details that I won't go into. Thank you. So now you see what happens, and you can see in the middle, as the red blood cell goes through, it takes a bullet shape and it springs back. The human eye cannot quite capture it. It takes 300 milliseconds for it to come back. It's a validation of the experimental technique. So this is the place where we started. Now let me go to the first disease that we will address, an infectious disease, a pretty lethal infectious disease, malaria, Plasmodium falciparum malaria. The history of malaria in the human body starts with the mosquito, the vector. When female Anopheles mosquitoes are pregnant, they need protein to nurture the eggs. They feed on human blood for the protein. If the person being bitten has the gamete for malaria, it goes to the gut of the mosquito, which processes it in the so-called sexual cycle of malaria in the mosquito, in the vector. When the mosquito feeds again on the human skin, she injects tiny particles called sporozoites, approximately 1 micrometer thick. Those sporozoites go to the human liver. The liver processes it over a course of 7 days or so, and then sends it into the bloodstream. That begins in the human body, in the bloodstream, a cycle, a so-called erythrocytic stage or a red blood cell stage, which lasts exactly 48 hours. So a particle or a precursor to a parasite, a merozoite, with a sharp tip called apical prominence, chemically targets the human red blood cell. It pokes into the red blood cell. So the particle is 1 micrometer. The red blood cell is 8 micrometer disk. It pokes into the cell. We have hemoglobin liquid inside in a soft sac. It gets inside the red blood cell. The cell seals itself. It's a beautiful engineering problem that we cannot mimic. We cannot mimic nature. And a 1 micrometer object pokes into an 8 micrometer disk. No fluid spills out. And the cell seals itself. And while the parasite is in the cell, it's in a cocoon. It's protected from the immune system of the body. Then it starts to transport proteins through the hemoglobin to the cytoskeleton, to the phospholipid bilayer membrane. And that causes geometric changes at the nanoscale. So in the first 24 hours, the so-called ring stage, changes take place. In the second stage, you form nanoscale knobs on top of the red blood cell. Then after about 36 hours, the cell becomes so stiff and so sticky that after 48 hours, the cell ruptures. A single parasite inside the red blood cell can multiply into many parasites, undergo nuclear division. When the parasites spill into the bloodstream, the immune system tries to fight back and raise the body temperature. So you know the febrile episodes in malaria, they are associated with this mechanical phenomenon of rupture. So when we take this, the question is, as the proteins get transported to the red blood cell, what are the changes in the properties of the red blood cell and what their implications are? According to the microbiology community, there are two major effects that are the pathogenic basis of malaria in humans. One is the stiffening of the cell. The other one is the increased stickiness of the cell. Both the stiffening and the stickiness make it difficult for the red blood cells to go through peripheral blood. So they can be cleared by the spleen if they can get to the spleen. And so that's one of the key reasons why in the severe forms of malaria, you get cerebral malaria, lack of oxygen to the brain. So trying to understand from a mechanical perspective these origins. So these two are both mechanical factors according to the microbiology community. At the time we started this work, based on instrumentation and tools that were available, the prevailing notion was the amount of stiffness of the red blood cell during the 48 hours was a factor of 2 to 3 compared to a healthy red blood cell. What I'm going to show you shortly is with more sophisticated advanced tools, what we find is that it's not a factor of 2 to 3, it's much more than a factor of 10. Even without understanding the underlying mechanisms, that has major implications. For example, some of the malaria patients also get drug therapy that is often given to stroke patients to improve blood flow. One of the drugs is pentaxiphyline. And what is the role of, what kind of dosage do you give to enhance flow to counter increase in deformability and cytoadherence has never been explored experimentally. So what kind of drug dosage do we give based on perceived effects in the human body, we can try to address with much more advanced techniques. So this is the motivation for us to take up malaria as our first study about 12 years ago. So we were very fortunate to work with the medical school at the National University of Singapore and also with Institut Pasteur in Paris, where I had the good fortune to spend a sabbatical in 2004 in the middle of a malaria group and hematology group. One of the benefits of that was right at that time, the Institut Pasteur people have successfully cloned the Plasmodium falciparum parasite, and they were also able to knock out, based on their field work in Africa, they were able to identify one particular protein called RISA, ring-infected erythrocyte surface antigen, from the parasite, and we could look at what is the role of that one protein based on gene knockout on deformability and stiffness. They were not only able to knock it out, they were able to knock it back in, which gave a very good control condition for a scientific experiment. So the experiment that I'm going to show here is the work of another PhD student, John Mills, who happens to come from Pittsburgh. He was my former graduate student. What you see here on the top is a healthy human red blood cell. And here is the challenge to do an experiment from a physics or an engineering point of view. You have a live human red blood cell that you have to keep alive in a phosphate buffered saline solution. And you have to immobilize the cell. You have to keep it alive. The cell diameter is microscale. It's 8 micrometers, or a tenth of the thickness of the human hair. But the force that you need to deform the cell to a level that actually happens in the human body as it goes through small blood vessels varies from 1 piconewton to about 100 to 150 piconewtons. So just to calibrate what a piconewton is, if you take an average size apple and hold it in your hand and stand on Earth, the force on your hand due to the weight of the apple is 1 Newton, named after Isaac Newton. A pico is 10 to the minus 12, which is 1,000th of 1 billionth of 1 Newton, the weight of an apple on your hand. That's the level of force. If the force was any higher, and if we have billions of red blood cells in the body, we'll feel it. The fact that we don't feel it, the force has to be very small. So the challenge in the experiment is, how do you do an experiment with a piconewton force, micrometer level displacement, and still keep the cell alive and make the measurement accurately? Fortunately for us, we resort to a technique well known in physics called laser optics or laser tweezers. This was the work for which former Energy Secretary Steve Chu got his Nobel Prize in 1997 in physics. So this we can apply to this. So what you see in the top, and using laser tweezers, I won't go into the technique, you take 2 high refractive index glass beads, functionalize them and attach them to diametrically opposite ends of a human red blood cell. They serve as handles, just like you do a material experiment. Then you can shine a laser beam on the 2 beads, and precisely you can trap the bead. If you move the laser beam, the bead will move with it. As you move the bead, you apply a force. And you use a standard tool called Stokes' law in mechanical engineering to calibrate the experiment. And this has been done over and over again in many different communities. We applied this to the case of malaria and human red blood cells. So in the top left figure is a live human red blood cell attached to the beads, no force. The middle figure in the top is a constant force of 68 plus or minus 10 piconewtons. You stretch the cell in a very controlled manner, and you can see how the red blood cell stretches. Then we increase the force to as high as 150 piconewtons, plus or minus 15 piconewtons, and we stretch it further, it stretches further. If you let the laser go, the cell will spring back to its original shape, as you saw in the video. The same healthy human red blood cells in the bottom figure, now you can see there are malaria parasites inside them. So the left figure, top and bottom, top is no parasite, bottom is with parasite, no force. The middle figure, 68 piconewtons in the top, 68 piconewtons in the bottom, except that the bottom one has a malaria parasite. Even with 68 piconewtons, it loses its ability to stretch. If you now move it to 150 piconewtons, it still has no ability to stretch. So we can do a lot of computational biology on this, and based on that, we can come up with predictions of what is the extent of stiffening or the reduction in deformability. So we found that it can be as high as 10 to 100, contrary to what was believed before based on cruder techniques, where it was believed it was maybe a factor of 2 or 3. So that was the first outcome of these experiments. So the next question is, what does it mean for, say, cerebral malaria? How can we demonstrate it? I mean, obviously, we can't image at the cell level at a single blood vessel in the human brain. What is the effect of this change in deformability on potential consequences for the progression of the disease? So this is an experiment that we did a few years ago. This is, again, a microfluidic device. It's a cocktail glass-shaped device with a long stem. So what you see, let me see if I can. I have a better laser pointer, so let me. So what you see here is not yet me. Thank you. Let me just stop. Stop there, that's fine. So what you see here, this particular cell and this particular cell each has a malaria parasite in this. Everything around it is healthy. So based on what I showed you earlier, these two cells are now too stiff because of the parasite. They cannot squeeze through the tube. All the other cells around them should be able to squeeze through. That's the hypothesis. Let's see if that works. Thank you. So you can see these two cells here, and we're going to add more cells around it shortly. Stop it. Can you click it again?
I think it should continue.
Okay, just leave it. Thank you. So you can see a whole bunch of cells. They can all go through except these two. Suppose you get 10 of these cells blocking an entryway to a particular blood vessel. The outcome could be very different. So there is no chemistry here. There is no biology here. Purely mechanics. Okay. So the next slide is, so we know that how much can you push this? So here is a sort of a dream question. Can you take a drop of blood, put it into a device that's about the size of your thumbnail, and just a drop of blood? Can you answer the following questions? Does somebody have malaria? Do they have fever because of malaria? Do they have Plasmodium falciparum malaria? Do they have Plasmodium vivax malaria? Because the mortality rates are very different. What is the parasitemia? What fraction of the red blood cells are infected with parasites quantitatively? Because that's an indicator of the quantitative indicator of the progression of the disease. What happened? Thank you. So this is, that's the question. Can we answer it with a microfluidic device? This is the work of a group of students and colleagues in electrical engineering that we published. Hans and Bo was a student I co-supervised who got so excited by this line of work. After getting a PhD in electrical engineering, he joined the Johns Hopkins Medical School to get an MD degree, and he'll be about 55 by the time he finishes all his training, but this is what he wants to do. So what I would like to show here is a series of experiments to address that question. So we have a diamond-shaped obstacle. We have a triangle-shaped obstacle with a converging flow or a diverging flow. We send live human red blood cells there. So the speed with which the cells go through the obstacles for a given pressure differential not only tells you whether you have malaria or not, it tells you whether you have Plasmodium falciparum malaria, which affects all red blood cells, or Plasmodium vivax malaria, which affects only young red blood cells called reticulocytes, which have been released by the bone marrow within 48 hours of release from the bone marrow. It also quantitatively can tell you how many cells are infected. purely from this obstacle course experiment. So, thank you. So this is an example of an obstacle course that you can create for red blood cells. And there is a lot of information. So the gist of this experiment is just what I summarized just now. So that's the first case that I want to demonstrate on the role of, in this case, physics and engineering in shedding light on cell-level disease for a infectious disease. I want to take a second problem. Again, physics plays a very important role. This is the work of a former graduate student who worked with me, Young Kyun Park, Paul Park, with me, and a professor of physics, the late Michael Feld. And this is in the spectroscopy lab at MIT. The work involves the following hypothesis. It's been known for 100 years that all living cells flicker, fluctuate, or vibrate. And that's been known through theory, through a variety of, they are chemically active, they are biologically active. In some cases, they are electrically active. So matter goes in and out of the cells, and they respond, they vibrate. And it's been postulated for a long time through work at Weizmann Institute in Israel, in Munich, Technical University of Munich, the New York Blood Center, and many other places, that the flickering, the level of flickering, is an indicator of health or disease at the cell level. Unfortunately, it was not possible to get full field. So the group at Narla Mohandas at the New York Blood Center, he did a series of wonderful experiments where he looked at one part of the cell and found that things like ATP, which is the chemical currency, can have a certain effect. And there is flickering. The group in Munich, which is also a highly reputable group, looked at it at a different part of the cell, and they found that there is flickering, but the effect of ATP is exactly the opposite of what Narla found. So both are correct, but their conclusions are somewhat mutually contradictory. So one is wrong, one is right. What is the technique? Part of the problem was neither group could look at full field. And here is the problem in doing a full field experiment, the entire cell. Again, you have to take an 8 micrometer cell, immobilize it, keep it alive in a saline solution. You have to have a non-contact experiment. The amount of membrane flickering is nanometers, so 10,000 times smaller than the thickness of the human hair. That's the amount of vibration. And the time scale of flickering is a millisecond. And you have to do the full field. And it's a geometry issue because it's a disk with a dimple in the middle. So how do you do this experiment? It turns out that physicists always knew how to do this experiment, except that it wasn't tried for the biological cell. So we decided, with having an exceptionally smart student, is that they quickly figure out how to do this. So here is a simple concept. I won't go into the details of this. And here is a simple concept. So if you send a laser beam of a certain wavelength, a so-called plane wave, and here is a human red blood cell that's immobilized, it has hemoglobin inside. The hemoglobin has a certain refractive index, just like a piece of glass, our eyeglass. Outside the cell, we have phosphate buffered saline solution, which has a different refractive index. So as a wave goes through the cell and then comes out, the difference in refractive index between inside the cell and outside the cell causes something called a phase shift. We can capture that electronically, digitally. That phase shift is captured by this function called delta phi, which we know from the experiment. We know the wavelength of light, and we know delta n, which is the difference in refractive index between inside and outside the cell. So if you know delta n, if you know the wavelength, if you know the phase shift, at any point you know the height of the cell or the thickness of the cell to a resolution of a nanometer. And you can do that for the whole cell, and you can do it at a millisecond time step. So problem solved. So we did that experiment. So the advantage would be that with this experiment, we could take a live human red blood cell, any cell, in this case a red blood cell, a healthy cell. We can shine a very low power laser beam on the cell outside the human body for a very short period of time, just from the membrane flickering, quantitatively, we could tell you whether the cell is a healthy cell or a disease cell. Now I can put a malaria parasite inside there. I can tell you whether the parasite is in the ring stage, trophoid stage, or a schizoid stage. And we can do that for other types of cell, non-contact. So I'm going to show you a video that demonstrates first for a healthy cell, then for an infected cell in the next slide. And so what you're going to see in this video is first the membrane flickering in a color contour for a cell. Then during the course of 48 hours, you'll see how the cell is being destroyed and how that's reflected in the flickering of the membrane. Thank you. So the top is the time clock in milliseconds. This is the flickering in height, vibration. This is the ring stage. That's the trophoid stage, late ring stage. And then the schizoid stage, which is close to 36 to 48 hours, you can see how the cell is destroyed. There is a lot more information in the published papers than what I'm presenting in the interest of time. So you can actually get the signature of these waves from the spacing in the spectroscopy signature. You can actually tell the length scale at which different proteins that tether the spectral network of the red blood cell to the phospholipid bilayer membrane are broken. And the reason you get a change in fluctuation is because different proteins are being slowly destroyed in the connecting points between the membrane and the cytoskeleton, and that's directly reflected, and you can quantitatively analyze this. We've also done experiments that I will not describe where you can actually add ATP or deplete ATP from the cell, and we can actually show that ATP has a huge effect. So now I go back to the controversy between one group talking about ATP having no effect, another group talking about ATP having a big effect, because they were looking at two different parts of the cell. When you look at the whole cell, you find that both are right. The techniques were right, the conclusions were right, except that they were looking at different parts of the cell. So that was resolved by this, and there is a lot more detail underlying this, which I won't get into in this talk. Now let me go to a very different disease, which is sickle cell anemia. So there is a whole range of complications that arise out of sickle cell disease. The main point is that in a normal hemoglobin, when you have globular molecules, you have a discocyte shape or a biconcave shape of the red blood cell. In the deoxygenated state, when the oxygen partial pressure is low, people with a sickle cell trait, they develop more of a linear molecular chain, a long inflexible chain that leads to the so-called classic sickle shape of the red blood cell. So you have multiple effects. The shape itself is not conducive to deformability. On top of it, you have stiffening of the cell that leads to changes in deformability. People with sickle cell disease, different densities that you can get within the molecular structure and the hemoglobin can lead to different responses. On top of it, you have increased cytoadherence, which also makes blood flow more difficult. The consequence is a variety of pain episodes, in some cases stroke, organ damage, swelling in hands and feet, and this is an example from the web that you can see in a child, what can happen because of sickle cell disease. So the question would be, what can some of these newer tools and technologies help with doing this? So you can look at it at different levels. People starting about 4 or 5 years ago started to look at that at macroscopic, tens of micrometer scale blood vessels. But a lot of the action takes place in this region where you need to go to smaller dimensions. So we hypothesize with respect to these experiments that under well-controlled transient hypoxia condition, the only drug that's approved, FDA-approved drug for this is hydroxyurea. So patients with hydroxyurea treatment and patients who have been off hydroxyurea, what are the kinetics of cell sickling and unsickling, and what are the rheological changes can we come up with better quantitative estimates of these episodes that patients face? So that's the objective of this. So one of the questions is, can you make the blood sickle in vitro? The sickle patient's blood sickle in vitro. So the next slide shows our experimental setup that was designed by our postdoc, Sarah Du, who's now on the faculty in University of Central Florida. So this is the device we built where we could control the oxygen partial pressure consistent with what happens in vivo. So we can make it sickle, unsickle, do the control timing, etc. We can do it with different drug treatments, so we can look at drug efficacy assays on the patient's blood, but outside the human body. We can do different therapeutic interventions through this. And these units are all in micrometers. So these are microchannels that we fabricated using the same technology that we use to make computer chips. So the next slide is a video, but before I start the video, let me describe what it is. So this is the blood sample from sickle cell patient. And initially in the video, you're going to see normal oxygen conditions. And a few seconds into the video, we'll slowly start to decrease the oxygen partial pressure. As the partial pressure decreases, you will see increased sickling of the cells. And then after some time, we'll slowly start to reintroduce oxygen, and you'll see the unsickling. And in fact, the time for unsickling, it turns out, is 1/10 of the time of sickling. And there are a lot of things one can do with respect to this. So this video now, we start with normal oxygen conditions. Now we take oxygen partial pressure. You can see, we reduce the partial pressure. You can see sickling now. So this is the patient's blood outside the body. It starts to sickle. And then in a few seconds, we're going to reintroduce, this is sped up, of course, this video. In a few seconds, we reintroduce oxygen, and it springs back to its original shape. So what is the consequence of that in the human body? I want to demonstrate that with the next video. Before I show the video, let me describe what this experiment is. So we create our obstacle course. So we rounded it off because there are sharp corners cause problems, damage to the cells. So we got cleverer after the first experiment, so we rounded this off. We send the red blood cells through here from the sickle cell patient. Now then we start to reduce oxygen partial pressure. And what you will see here is what happens. Cells can go through this quite a bit when the oxygen level is, when you have normoxia conditions. When you have hypoxia condition, the ability of the cell to go through these obstacle courses significantly diminishes. So that's shown in this video clip. So you can see cells going through without any problem under normal oxygen conditions. By the way, we benefited quite a bit from Dr. Greg Cato, who's a hematologist at Pitt and UPMC, in this collaboration over the last few years. And you'll see slowly, one by one, they start to get obstructed even at the entryway, and you'll see in a few seconds, more and more of this happens in this experiment. This is in vitro. And so this gives us a tool. We are actually working with several different hospitals around the country on different, for example, controlling hypotonicity for the red blood cell, adding water. There are a lot of these tools have been around for quite some time. How effective are they really? And can we quantify them with these kinds of experiments? So there are a number of experiments that are ongoing right now with respect to these particular experiments. So one of the things we can get out of these experiments is delay time of cell sickling versus density fraction from the patient's blood, and also sickle fraction of hydroxyurea on hydroxyurea and other kinds of therapeutic interventions. So this is just a summary. There is a lot more detail that I won't go into. Let me come to the last few minutes of my talk and finish with two examples. So going from the cell level now to the organ level, one of the organs in the human body that's affected by cell deformability, which is sort of a filter for defective cells, especially red blood cells, is the spleen. And whether it's malaria or sickle cell disease, the mechanics of the spleen plays a huge role. And it's a very complex topic, but the spleen has the smallest fitness test, the most stringent fitness test for the human red blood cell in terms of the so-called interendothelial slit between endothelial cells in the spleen. I won't go into the geometry, but you can model that using computational biology tools in a very rigorous way. But I'm going to show you an experiment that is being done jointly between Institut Pasteur in Paris and our group. And the experiment is the following. Dr. Pierre Buffet, who's a scientist physician, he does isolated perfused human spleen systems. When somebody has benign pancreatic tumors, in order for the surgeon to access the pancreas, in some cases, you need to remove the spleen. When you have access to the fully functioning normal human spleen, you can do a number of experiments with the spleen outside the human body in the ex vivo experiment. What Pierre and his group have done over the last 4 or 5 years is not only to study things like how the malaria parasites go through the live human spleen outside the body and use our computational biology tools to calibrate their experiments. For example, we know the blood pressure, we know the pumping of the heart. What is the pressure differential ahead of and behind a cell, a red blood cell as it goes through the body? What is the pressure differential in the human spleen? How does it pass the fitness test? We have no quantitative understanding of it. So through Pierre's experiments, ex vivo experiments on real human spleen, and our experiments and computational models, we've been able to guide their not only work in this area, but they've also developed and patented a synthetic spleen. A synthetic spleen made of 2 metal beads. And the beads, they've run experiments with malaria parasites with a synthetic spleen, with a human spleen. So we have connected that with our experiments of their synthetic spleen so that we can guide the design of their synthetic spleen. We've also done a computational 3-dimensional model of the real human spleen. So if you could show the top image. So this is color-coded for strain contours. And you can see how the cell goes through the synthetic spleen, a live human red blood cell. And the bottom right figure, if you click on that, thank you. That shows how the red blood cell is likely to go through the interendothelial slit. This brings to one of the most fundamental questions of nature. There was a paper published in 1968, a wonderful paper by 2 people, Carnegie and Burton, that claimed that the volume and area of the human red blood cell is determined by the smallest blood vessel in the human body. So they measured the surface area of a red blood cell. They measured a volume of a red blood cell for large numbers of patients, healthy people with no diseases. And they came up with a theory and a model showing that the reason the area to volume ratio of a red blood cell is a certain ratio is because it's determined by the smallest opening in the human body. So this was in the 1960s. They didn't have the benefit of the tools that we have or the computational power that we have. So we decided a couple of years ago, because now we have experiments on the real human spleen, we have computational tools, we have all kinds of wonderful nano tools, we can actually ask if that is really true. So last year, we published this paper together. So this is the work. All these points here are the data points that Carnegie and Burton published in the 1960s. About 20 years later, another group, Gifford et al., came and published and showed that the surface area of a healthy red blood cell, the volume of a healthy red blood cell, should follow this rule. Then they claimed that the determining factor for the area to volume ratio for a human cell, red blood cell, in a healthy human, is determined by the small blood vessel. So we decided to put that to test using a simple mathematical model where we could predict the interendothelial slit as the determining factor. and showed in this paper last year that it's not the smallest blood vessel, it's actually the slit in the spleen that determines the area to surface ratio. So this is the experimental data from their work. The dashed line is this equation, this set of equations, mainly this equation. And all this purple color is a fully three-dimensional computational model called dissipative particle dynamics, done in collaboration with a group of people at Brown University in their math department, and my group and a group at MIT. So you can see that this sort of provides a bound. Now you can take it to other diseases. So if you have sickle cell blood, it should fall outside of this range. If you have spherocytosis, if you have elliptocytosis, if you have Asian ovulocytosis, those diseases should be abnormal. So this equation and this computational tool now provides a guidepost on surface to volume ratio as a marker for potential. One more factor in disease diagnostics. I just want to make a statement with respect to this. So this paper that was published in the National Academy of Sciences Proceedings last year, this is in relation to the editorial, the op-ed that I had yesterday, or the letter to the community that Art was mentioning. I just want to tell you where all these people came from. Igor Pivkin is a Russian mathematician who got his PhD at Brown University in applied mathematics. He now works in the Bioinformatics Institute in Basel, Switzerland. Zhangli Peng came from China, did a PhD in Texas, did a postdoc at MIT. He's a professor at Notre Dame. George Kaniadakis came from Greece. He's a professor of mathematics at Brown. Pierre Buffet is a French clinician scientist working in Paris. Ming Dao came from Shanghai, and I came from India. This is the team that has worked on it, and we work on in 4 different countries right now. I just want to make that point about how science progresses. And the last example with which I'll finish relates, again, bringing science and engineering for a completely different problem, my third disease class, which is isolating a rare circulating tumor cell in the human body. So this work has benefited enormously from collaboration with Professor Tony Huang, who until recently was a faculty member at Penn State University, and since last July, he's been a faculty member at Duke in their biomedical program in North Carolina, and Ming. So let me explain the rationale for this work. As many of you in the audience know better than I do, there are so many clinical applications with respect to circulating tumor cells are a very, very complex topic. Their shapes are different, their properties are different, and what you do with them and how well you capture them, and sometimes it's like finding a needle in a haystack. So the intention of the experiments that I'm going to describe is not to change current medical practice, but if we can add one more tool to the list of options that's available to a clinician or a cancer doctor or a patient, we probably increase the odds of somebody surviving a few more years. That's the spirit in which these experiments are done. Potential applications of being able to capture them and also capture them unaffected will lead to early cancer diagnosis, real-time disease progression monitoring, tumor staging criteria, personalized drug treatments and susceptibility tests, and treatment targets. About a year ago, there were 305 clinical trials in the US. There are probably much more now. The 4 major types of experiments that are being used around the world very broadly fall into the following categories. Centrifugation, where essentially you spin and then try to isolate mechanically. Of course, it's a high G-force, and that can cause damage to the cell and its properties. FACS, or fluorescence-activated cell sorting, is another technique, a tagging technique. Magnetic beads, and then different types of labeling or filtration, whether it's electrical, chemical, biological, or just mechanical filtration through a pore. All of these have their benefits. They also have their drawbacks. But one of the critical factors is that if you capture a cell, however you capture it, are you altering the properties of the cell because of the method you use to capture the cell. In other words, the artifact of the experiment. All of these techniques have artifacts. So for a practicing clinician or somebody who diagnoses the disease, it's very important to know what the artifacts are. So we decided that because ultrasound is so commonly used, hundreds of millions of times all over the world for many, many decades, safely on humans, can we take ultrasound as a technique at the cell level so that we can actually capture the cell? So the technique involves the following. We use something called standing surface acoustic wave. So essentially, there are some things called IDT, interdigitated transducers, which are in our cell phone. And there are two IDTs. So this is a US dime. So the marker, these are the IDTs. This is the device that we are using compared to 10 cents. So that's the size of the dime to calibrate it. So these IDTs send out surface waves. So if you have a fluid flowing through, let's say first the fluid has particles in it, say plastic particles. And as the fluid flows through, as you generate a surface wave, it causes what is called a radiation pressure on the particle. In this case, say a polymer particle. So you have four forces. You're pushing the fluid with a pressure, so there is a forward force. There is a drag force that's preventing the fluid drag that prevents the fluid from flowing. You have something called the buoyancy force that tries to lift the particle, and you have the gravitational force that tries to pull the particle down. For a microfluidic device, the buoyancy force almost exactly balances out the gravitational force. So only in the horizontal direction, you have forces, net forces. So if the fluid is flowing in this direction, if you have a particle, the particle gets something called radiation pressure. So now I can do two control experiments. I can take two particles of the same material, but slightly different size. So I can take a slightly smaller particle, let's say 8 microns or 3 microns, and a 5 or 10 micron particle, send them together. The two particles will experience two different forces from the acoustic wave. The larger particle will go in one direction, the smaller particle will go another direction. I can separate them. Now I can do the following experiment. I have two particles of the same size, but slightly different material. The physical properties are different. Because the physical properties are different, the radiation pressure will be different. And therefore, one particle will go in one direction, another particle will go in another direction. I can separate them. So we did that. We did that with an efficiency of more than 95%. So once we did that, then we thought, okay, if that is the case, we can do something better. If I have healthy white blood cells and you have cancer cells, there is both a difference in size and different property. I should be able to separate the cancer cell from the red blood cell. So we first started with cultured cells to see if the experiment works and the hypothesis works. So we took breast cancer cells, cultured cells like MCF7, and we took cervical cancer cells, HeLa cells, and we spiked the concentration, 50% healthy cells, 50% cancer cells. We mixed them, sent them through this. We could separate the cancer cells from the healthy cells quite accurately. Still, it doesn't solve the problem because it doesn't solve the problem of circulating tumor cell because you want to find 1 in 100,000 cell. Then we went to Hershey Hospital in Pennsylvania because we had some initial connections and collaborations there. And we got blood samples from 3 breast cancer patients. And we took blood from the patients and did a series of experiments. Of course, they diagnosed the cancer using other techniques, and we were able to capture the circulating tumor cells out of this, and we could capture 1 in 100,000 cells. Still, the technique is not accurate enough for prime time. It's still primitive. It's still at a lab scale. It's not ready for prime time, but this is what we demonstrated last year. So this is essentially the technique. Let me show you the video. So here you see in the top you have cancer cells. This is the cultured cells. You have cancer cells going this way, healthy white blood cells going in this direction. We can turn the surface acoustic wave on or off, and we get different results. Here are the examples of various experiments we did in collaboration with Hershey Hospital. And this is just last year and a half. These are the breast cancer cells, HeLa cells, etc. We were able to successfully, these are the number of cells we were able to capture, how many, 1 in 40,000 cells, 1 in 100,000 cells we were able to capture. Still, it may not be good enough. But equally, we've been able to show that we are able to capture these cells, and these cells, at least based on the experiments we have so far, I cannot confirm that 100%, based on the experiments we have so far, it seems that the cells are not affected by our experimental technique because the acoustic wave is benign on the cell. It doesn't alter the properties of the cell. There is no other contact that's involved here, unlike the other techniques that I described. So this remains to be seen. So with that, I want to conclude with a few observations. So hopefully I've shown through a variety of examples that we have possibilities for understanding human diseases at the cell level using a variety of experimental and computational tools, in some cases at the organ level. We have potential benefits, fundamental mechanistic understanding, diagnostic capabilities, potential for novel therapeutics. And this is a field where 15 years ago, most of the bioengineering departments around the country, including at Pitt and Carnegie Mellon, they started about 15, 20 years ago to come into full swing. And in the last 15 years since we started our first work, the number of people working in this kind of a field at the intersections has really grown exponentially. And hopefully this will bring together hitherto disparate communities in ways in which we have not seen. And there is quite a good possibility for assessed drug efficacy assays as well. As I mentioned, this work would not have been possible without collaboration from a large number of groups. This doesn't even do justice to all the people from whom this work has benefited. First and foremost, I want to acknowledge Ming for being part of all of this work. A number of people at CMU have been engaged in different aspects of this work. A large group over the last 10, 15 years in Boston, at UPMC and Pitt. I mentioned Adrian Lee, working with him on the CTC part of this. Because Yoel Sadowski in a variety of, especially with respect to acoustic waves and its implications for placenta research. Greg Cato from hematology, Tony Huang, George Kaniadakis at Brown University. We benefited enormously from the medical school, both at the National University of Singapore and Nanyang Technological University. Without their help, this work would not have been possible 10 years ago. And so a number of folks have helped us in this work. Institut Pasteur in Paris has been an absolute necessity for us, and it's a wonderful group of colleagues with whom we have benefited. And most of this work has been supported by NIH, and my work is not supported by NSF because I've been barred from applying for grants from NSF. This is the punishment for public service. So I want to thank you for your attention. Thank you very much.
Well, because of the lateness of the hour, we're going to hold a reception for Subra up in the lobby, and those of you who have questions or comments can have at him there. But first, before everybody leaves, we have a gift for Subra. So, the gift is a medical school chair.
Our daughter is an MD who's now in pediatrics in New York City, and she calls me the useless kind of doctor. So I couldn't do 1,000th of what she's able to do. So
let's all go upstairs and celebrate Subra.